Okay. And anybody would like to tell us about something about digital microscopy? What do you understand by pixel? You talked a lot about pixel yesterday. What is a pixel? Okay. So, ask this question. That's good. So pixel is basically the word. If you see the pixel word comes from picture elements. So every picture has an element that, like we were talking yesterday, 0 and 1. So every point in the picture has to be characterized. So depending upon the number of pixels, you have the quality of the image. That is what it was being taught yesterday about JPEG format, TIFF format. So we'll keep on talking about it. Let us start the session and then. So I invite Dr. Amita Aggarwal, Head of the Department of Clinical Immunology. Ma'am has been working and talking about, it is really nice to listen to her. She'll be talking about fluorescent microscopy. And this is after the light microscopy that we now move on to the different specialized techniques of microscopy. Thank you.
Thank you, Vinita. And good afternoon, friends. And how many of you use microscopes? Okay, maybe the other way around. How many of you have never used microscope or don't use it regularly? A lot of you. So when you were students of 12th class or 10th class, that time you were using microscope in the lab or during your pre-medical. What was that type of microscope? Please come. Any idea what are the different types of microscopes if you have been taught last time, yesterday? Anybody? Bright field microscopy. What else? Electron microscopy. Fluorescent microscopy is written there. Confocal microscopy. Anything else? Polarizing microscopes. Which one? Atomic force microscopy. Okay. Where did you hear that word? Atomic force microscopy. So anybody else? Anything more? People who do cell culture? Anybody of you do cell culture? What microscope do you use? Inverted phase microscopy. So we have many different types of microscopes and each one has different function. And we are basically trying to visualize mostly cells. And for some help us to see live cells, some help us to see cells in their original without any staining. In some we need to stain. Fluorescent microscopy is one different form of microscopy where we are utilizing a chemical principle of fluorescence to see that are there certain proteins present or not. So the next question, this should not be answered by people from immunology at all. So tell us, what is luminescence? Anybody has an idea? If you have read physics during your 11th and hopefully the 12th grade and mugging up those MCQ questions, what is luminescence? Now you have to put on your mic and speak. Yes, just switch it on for me.
Ability to emit the light.
Ability to emit light is luminescence. Okay. So can you give us an example of luminescence in daily life? Anybody? Firefly. Yes, firefly is a luminescent animal. So what is fluorescence then? Is it type of a luminescence or is it something different? Now you don't speak to yourself, speak into the mic so that everybody can learn from your knowledge. Anybody who can tell us what is fluorescence? Yes.
Absorbing the smaller wavelength of light and emitting the larger wavelength of light.
Then what is the difference between phosphorescence and fluorescence? These are simple things we read in chemistry, you know. You must have heard example of phosphorus. Phosphorus is a phosphorescent compound. Is there any difference or no difference? How many of you think that there is no difference between phosphorescence and fluorescence? There is a difference. So what is the difference, dear?
The difference is that phosphorescence continues to be happening even if the light source is removed.
Okay.
And fluorescence is till that source is being emits light, excites.
So you are fairly correct, not exactly correct. So let's see the definitions. So as somebody said, luminescence is any emission of light, and light we know is an electromagnetic wave from a substance that does not arise from heating. Because when we heat something and then when you get light, that is called what? Suppose you heat wax, you get light. Do you call it luminescence? No. So that is called, if you can read for yourself, incandescence. That is when you get light after heating. So it is different from that, and it generally happens when the electrons go into the outer orbit, and when they return to their original orbit to be in stable phase, they will emit energy. Some of this energy is emitted as light. And as he was saying, sorry, I didn't get your name. What's your name? So as Anubhav said, that fluorescence is emission of this light energy or the electromagnetic radiation, which is usually in the light visible range. It's caused by excitation of atoms going into the outer orbit, and which then re-emits almost immediately. He was trying to say that when you have the source going of excitation, the same time you have the emission, but there can be a minor time lapse, but it does not last for long periods of time. In contrast, as Anubhav said very clearly, that phosphorescence is a property where the emission can go on for a long period of time. And it may not happen immediately as soon as the excitation happens. It can happen after a lag period. So that is a difference between phosphorescence and fluorescence. And obviously in lab, we don't want to wait. We are not going to use phosphorescent compounds. We are interested in seeing something. As soon as we excite, we want to see under the microscope. You're not going to wait there for minutes to light to get emitted. So that is why when we want to do this, we use fluorescent compounds. And this is what we are doing. When we are exciting, you are making the electrons in the outer orbit to move into the next orbit. And that is an unstable thing for any chemical when the electrons are not in the regular orbit. So when they come back to their original state or the steady state, you will have some energy being released as light and some energy being released as heat. And as you know that every chemical reaction, when it happens, if you like when we were 10th class or 11th class students, we used to do those color reactions in the test tubes. The test tube would get hot whenever you added something to it or a chemical reaction happened. So every chemical reaction is partly a thermal reaction. So there will be some energy produced as heat. Because some of the energy is being lost as heat. That is why the energy of the emitted light is less or more than the excitation light. Less. And what is the relationship between wavelength and energy? Inversely related. As that girl has said, that the emitted light has a higher wavelength than the excitation light. So you realize that all what you read during your life is important every time. You can't afford to forget these basic principles of physics and chemistry. And this is what it is showing, that it emits light of lower energy and a higher wavelength. and this is what you see. If you will see any fluorescent compound, if you want to go and buy, this is what will be shown in the sheet of that compound. That what is the absorption maxima or the excitation maxima of that compound, and what is the emission maxima. At what wavelength does it emit light, and at what wavelength does it get excited. So you need to know these when you choose the compound which you want to use in your experiment. So what is the most commonly used fluorochrome? Anybody? People who use microscope said, so many people were using microscopy. Any one of you use fluorescent microscopy? Anybody? Yes. So which fluorochrome are you using? Just put mic on.
Tapping FITC.
DAPI, FITC. So what is the excitation maxima for DAPI?
I don't remember.
You don't remember.
Yeah.
What is the emission maxima for DAPI?
I know FITC. It's 529.
590 is the
529.
529
29.
is what? There can't be generally one. It's
Yeah, it's the
as you
emission.
seen here, no, it's a spectrum.
Yeah, it's an emission spectra, 529.
Mhm.
And I guess 428 is the excitation.
Anybody else wants to improve? Yes, Shivika.
Ma'am, it's 488
488 is what?
excitation.
488 is excitation maxima, and emission.
Ma'am, near about 550. 550.
Yeah, so it's about that. So generally 488 is the red area. So generally when we do flow cytometry, you use red laser. Red laser is the most commonly used laser. Red or blue?
Blue.
I said blue, sorry. It's blue laser. So blue laser gives you excitation at around 490, 488, 490. And most of the compounds which were initially developed were the ones which could get excited at around 480 to 490, because the blue laser was the initial source of lamps to excite this. So you are right, for FITC 590. So this is giving you some idea of what it is. So if you see, lot more compounds are getting excited at 480 to 490. That is the blue area, and FITC is around 515, 520. Phycoerythrin gives you red color, so it's around 580. Ecridin orange is also gives you orangish, yellow, orange color, so it's 510. And PI gives you at around 630, 620 to 630. So this is important to know if you want to combine two compounds. In the same slide, you want to use two different fluorochromes. So then you need to know that they should get excited by the same light, but they should emit as wide as possible, so that you can differentiate those two colors when you do 2-color, 3-color, 4-color fluorescent microscopy. It is possible to do multiple color fluorescent microscopy. So this is what I was asking you, that what are the two different kinds of microscopes? This is what we used to see in school. We used to have that lamp, or some people used to have that reflector there. You take the sunlight, reflect the light comes there on the reflector, then it goes, there's no pointer. So the light used to get reflected and used to reach the stage. On the stage we had the slide. The light would pass through it, and then we will see it in the eyepiece. These are the microscopes we have seen in the routine schools and colleges. And probably these are still the same when we use ordinary white light microscopy. So what is the problem with this microscope? When you have, these are called transmitted light microscope, that the light is coming from below and it is passing through and through your slide. What happens? If you remember principles of light, the principle of reflection, transmission, and what is the third one? Refraction. So what happens in this? You must be wondering, we have reached a physics class. So when you have light going through and through, and these are generally glass slides, some amount of refraction would occur. The second, because of that, you get diffraction of light. So the image which is formed is not very sharp. And as you must have seen in physics, the principle of reflection is perfect. If light is coming at 90 degree, it will go at 90 degree. If it is coming at 120, it will go at 120. There is not even 1 degree of diffraction. So what people have tried for fluorescent microscopy, because the amount of light is very little, you don't want to lose any signal. So you want all the signal to come exactly like that. So that is why most microscopes which are used nowadays are epi-fluorescent microscopes, where if you see, light is coming from top. Light is coming and getting reflected by this mirror, dichroic mirror. It will come down, it will hit your object and will get reflected without any diffraction, because it is a principle of reflection. So you get absolutely sharp image with low intensity of light. So what do you think this mirror is? A mirror, does any mirror allow light to pass through? Have you heard of any mirror which is allowing light to go through it? The mirror are supposed to be only reflectors, no? They only reflect light. So these microscopes have this special mirrors. These are called dichroic mirrors. So these are lenses plus mirror. So they have a property of reflecting light and also allowing light to pass through. Actually, this is the major advance which happened, which led to development of flow cytometry, which you probably may discuss later. So it will allow certain wavelength light to get reflected and a wavelength which is higher than that to pass through. So we said that the light which is being produced is of a higher wavelength. So that will pass through while the rest of the light will get cut. So this is the diagram of a epi-fluorescent microscope. So you have the lamp source, which initially used to be halogen lamps and nowadays are LED lamps. So to have brightness of light, and the light comes here. This is your dichroic mirror, which will reflect the light to the stage, and then the reflected light goes through this and you can see it through the eyepiece. And if you see, these are the kind of microscopes which are available, which have become very small, because LED source is a very small source. And with advances in computing, you have nice screens. You don't have to strain your eyes. You can directly see on the TV screen by putting a CCD camera there, and you can capture your images and store them. So when you do any assay in immunology, all of them have either direct, you must have heard of Coombs test, direct Coombs test, indirect Coombs test. Similarly for fluorescence, direct immunofluorescence, indirect immunofluorescence. Direct means it's a one-step assay. A one-step assay is that you're looking at something which is bound already, that the antigen-antibody interaction has already happened, or the antigen is already deposited. So you add an antibody to that, which is labeled with a fluorochrome. If that protein is present, that antibody will bind. And then if you will put light on it, you will see fluorescence. So it can be, it has multiple functions, especially in clinical medicine. Direct immunofluorescence assay is very often used, and we'll see in which situations. So skin biopsies to diagnose immunologically mediated skin diseases like bullous pemphigoid, pemphigus vulgaris. I'll show you some pictures. And the most important is kidney biopsy. Because when a patient comes with glomerulonephritis, you don't know what type of a glomerulonephritis the patient has. So doing the immunofluorescence on the kidney tissues tells you the 3 different types of glomerulonephritis which you can develop. So this is the skin biopsy pictures. So what you see here, you can see this is the epidermis, and underneath this is the dermis. So you have fluorescence at the level of dermoepidermal junction. So if you are tagging anti-IgG, looking for IgG deposit, that the anti-IgG antibody is tagged with the fluorochrome, this can happen in patients with lupus. If you are using anti-IgA antibodies which are tagged with fluorochrome, then generally we use anti-IgA antibodies in patients with dermatitis herpetiformis. They have anti-IgA antibodies deposited at the dermoepidermal junction. So is there any clinician here? Anybody who's from clinical medicine? None. How can that be? So in this course, no clinician? You're from which department? Nephrology. Oh, you're the best person. Tell us, what is this? This is skin biopsy. Which one? Which one? Which one is Boothler's pemphigoid? 1, 2, 3, 4. Left top and left bottom are same, dear. Okay, so you see here, what are you seeing? This is the epidermis. Epidermis is showing you that around the keratinocyte there is fluorescence. So this fluorescence is in between the cells. So this is also called crazy pigment or something. So this is in patients with pemphigus. And I told you about dual color fluorescence. So here you have stained the nuclei with DAPI, which is giving you blue color of the nuclei to identify the cell. And this is happening at because the antibody in pemphigus is against desmogelin free, which is intercellular substrates. So that is why you are getting this kind of a pattern. So for you, this is the bottom one is the pemphigoid. This one is the pemphigoid. In pemphigoid also, you get dermoepidermal fluorescence, but there is some amount of fluorescence which is also seen in the epidermis. So this is lupus band test. This is pemphigoid. This is pemphigus, and this is just to show you dual color fluorescence with the nuclei stained with DAPI. Okay, kidney biopsy. Your friend has arrived now. You are also from nephrology? No. Are you a clinician? Yes. Tell us, what is the usage of immunofluorescence in kidney biopsies? You can put on the mic from there, if the mic is there. Okay, otherwise you can tell us just like that. Are you in medicine, medical specialties or surgical? Surgical. Surgical people don't remember this stuff. Anybody from medical specialties? None. That's very odd. Are you from which department, dear? Last bench? Surgical gastroenterology. All of you? Yes. Endocrinology, you tell us. You must have read in the MD medicine, no? What is the utility of doing immunofluorescence on kidney tissue? Okay, so the major issue when we do immunofluorescence on the kidney biopsy is, as you were saying, we look for IgG, IgA, IgM, complement, and fibrin. So generally we do for 5. If you find everything there, it's most likely lupus. If you find only IgA or a little bit of IgM, we say it's more likely to be IgA nephropathy. And there are many more kind of things. The other way to look at is that what is the kind of deposit? So if you see here, you see around the glomerular capillaries, you are having this kind of a granular staining. When you get this kind of a granular staining, it is most likely that there are immune complex deposited, which are little big in size. In contrast, you see this. It is like as if you've taken a pencil and drawn around it. So this is what you see in anti-GBM disease. In anti-GBM disease, it's a linear deposit. While in immune complex mediated glomerulonephritis, you get these kind of a granular deposit. Another thing you can see here is the crescent. And sometimes even the tubules can show you immune complex deposits, which are against the tubular basement membrane proteins and other things. So kidney biopsy, actually every kidney tissue removed at kidney biopsy should be subjected to immunofluorescence because it helps you to find out, is it an immune complex mediated glomerulonephritis? Is it posse-immune or is it anti-GBM disease? Then comes the indirect immunofluorescence. Indirect immunofluorescence, like indirect Coombs test, is that you are looking for pre-existing antibodies in the serum or in some fluid, CSF, pleural fluid, anything. So here what we need two things. One, we need a substrate which contains the antigen against which we are seeing antibodies. So if you want to look at anti-nuclear antibody, you must have some tissue which has the nuclei. And second, you need to have antibody to your antibody or anti-immunoglobulin, which is labeled with a fluorochrome. So that when you will put light, you will be able to see fluorescence. So these are useful for detecting antibodies in the serum. And this is what we do in ANA. You have these spots which have the cells. Patient sample has got these anti-nuclear antibodies. You put the patient serum, incubate for some time. The antibody will bind to this antigen. Then you add fluorochrome labeled anti-IgG antibodies. Then you wash and then you see under the microscope. So you can see different patterns. And these patterns just tell us that what is the likely source of antigen. Against what antigen is this antibody likely to be? It may not be 1:1 relationship. So this is what it looks like in real life, that you can have pattern which is absolutely homogeneous. You can have rim pattern, you can have specks, and you can have pattern which stains the centromeres of the cell. Then the other antibody which is most often ordered by many departments is ANCA, that is anti-neutrophil cytoplasmic antibody. Because it is against the neutrophils, so you need to have the neutrophils there. So you have smears of neutrophils, you add patient serum, and then after adding the conjugate, you look for this. So you can have many different patterns of anti-neutrophil cytoplasmic antibodies. And then we can look for many other antibodies. So this is for gastroenterologist. You look for anti-mitochondrial antibodies. So you need to have many tissues. So you have a composite block which has liver, kidney, stomach, and everything. If you see fluorescence pattern like this, which most likely represents that it is staining the mitochondria, then we say that it is anti-mitochondrial antibodies. If you find staining of the smooth muscles, you say anti-smooth muscle antibodies. So the advantage of fluorescent microscopy is that it tells you which tissue is getting stained. That will tell you which antigen, against which antigen is this antibody. So that is an advantage of using fluorescent microscopy. And this is for liver kidney microsomes. Because here certain tubules will get stained, while in anti-mitochondrial all the tubules were stained. So that helps us differentiate, is it AMA or LKM antibodies. Then in research, sometimes we also use avidin biotin system to amplify the signal. If the signal is very weak, you're not able to see. So then you add biotinylated antibody, because each biotin molecule can bind 4 avidin molecules. So your signal gets multiplied 4 times. And that can also be utilized. So these are, because we have this is a course on microscopy. So you need to know that how you can improve your quality of images. The quality of image can be improved if you use good quality reagents and you have a good quality microscope. So what is a good quality reagent for a fluorescent microscopy? Is that you must have optimum amount of fluorochrome attached to your protein. And so what we call as fluorescein to protein ratio. Ideally, it should be between 1 and 6. If you have too many fluorescent molecules attached to your anti-IgG or something, then you'll get a lot of background staining. So your noise will increase. If you have too less, then your signal will be too weak. So you need to titrate your antibody concentration also. Don't add too much of antibody. But nowadays, most protocols will tell you how much is needed. You can tweak it a little bit, but generally it works in the range which is written in the kit or something. The issue of non-specific binding is always there because you are adding a protein tag to a fluorescent tag. And when you suppose sometimes what happens is the fluorescent tag has come off. So you always spin the conjugate before you add. Otherwise, there will be specks of fluorescein lying all over your slide and you won't be able to interpret properly. Heterologous antibodies will give you cross-reactive antibodies can sometimes give you a non-specific pattern. If you don't do proper washing, loosely bound antibodies to some other antigens can also give you a non-specific binding. So your washing has to be perfect. And obviously certain tissues have this property of yes, question. So heterologous antibodies there are many kind of antibody. One is a heterophile antibody or heterologous antibody which form during certain viral infections. These will bind to many proteins. So they will give you a false positivity in almost all tissues which you examine. So then you think that the patient probably has had some viral infection. And those have no specificity to any tissue at all. Cross-reactive is that the antigen which you are testing is pretty similar to the antigen against which that antibody is supposed to be. So there you will not get a very non-specific binding, but you may get in certain patients or certain tissues may show you binding in that. That's how you differentiate, is it a heterologous antibody or is it a cross-reactive antigen? So the other problem is natural fluorescence of certain tissue, like neutrophils. Neutrophils have so many granules. So when you do ANCA or anti-neutrophil cytoplasmic antibodies, even if you don't put any fluorescent tag, you just put the neutrophil smear in the fluorescent microscope, there will be some amount of fluorescence seen because these granules contain a lot of azerophilic material and granules are there. So that you have to see. And second, if you use formalin fixed smears, so that's why for fluorescent microscopy, formalin fixed tissue is not utilized. It will give you too much of autofluorescence. Then, as I said that you need to always run positive and negative controls. You need to know what is your signal and what is your noise. The background staining. That will be the negative control will tell you what is the level of background staining. It's never black. It's always some amount of staining will always be there because of non-specific binding. So always run positive and negative controls in every slide. And second, as I said, that not only your chemicals or reagents, your microscope also has to be of good quality. So there are quality slides available, optical standard slides, which tells you is your microscope properly aligned, are you getting adequate light, everything you have to see. Is the filter which you are using is correct or not? So if you have everything optimum, you will get the best quality pictures. Then other applications, so these are the ones which are most often used, which I discussed. That is the direct fluorescence, indirect fluorescence for looking for autoantibodies or deposited immunoglobulins. But fluorescent microscopy can also be used for detection of microbes. Can somebody give an example? We have some people from microbiology here. Who's from microbiology? Nobody? Oh, two hands. Tell us. Two applications. Where do you use fluorescent microscopy for looking for a microbe? Treponema pallidum. Yes. Anybody? Which viruses? CMV. Generally, we don't use fluorescence. Nowadays, it's all PCR. So the classical is Treponema pallidum. And if you see, let's first see Treponema pallidum, how it looks. It looks so beautiful. It's a spiral shaped organism. So you can see how good it looks. But it can be utilized for anything actually. If you have a good antibody, which is fluorescent tag, people have used it for chlamydia, people have used it for viruses, people have used it for parasite, anything can be used. Mycobacteria, that's what I'm saying, that it can be utilized for any of these. Looking for in a opportunistic infections, cryptosporidia, microspora. So this one is showing you cryptosporidium. So you can utilize for, it gives you very beautiful pictures and specificity. So here it's being used for, in the tissues also you can see. So this is for rabies, virus in the brain. So then for genetics. So who is from genetics here? Fish. Anybody of you done fish? Pathology? Do you do fish? Fluorescent in situ hybridization? That's also a fluorescent microscopy. You use the same microscope which I showed you. So here we are looking at, you have the cells on the slide, you cause denaturation, your DNA becomes separated out, and then you hybridize by using this probe which has got deoxygenin on it. And then you add antibodies which are labeled with a fluorochrome, anti-deoxygenin antibodies. And then you look for it. So can anybody tell us one of the simplest example of fish in clinical practice? Anybody? Which one? Mic में बता सुनता नहीं. HER2 new. Are you doing fish for that? Okay. So maybe when we trained, Bcr Abl was the most earliest usage of fish was in Bcr Abl translocation seen in patients with CML, because the patient's peripheral blood would have so many cells. You just make a peripheral smear, or you can do if you have done a bone marrow exam. So here the ABL is labeled with red dye and the BCR is labeled with, antibodies to BCR is labeled with green dye. So normally they are located on two different chromosomes, 9 and 22. When this fusion happens or translocation happens, then they come together, and you can see so beautifully where the arrow is given that in these cells there is Bcr Abl translocation. So when this imatinib drug came, people were looking out how many of them have had cytogenetic remission. So you can count the number of cells which have this Bcr Abl translocation. If the numbers have come down, that you know the patients have had, besides the clinical remission, there is also a cytogenetic remission. You can also do RNA fish. There I showed you was a DNA fish. You can also amplify RNA. You can label the RNA, use a probe which will bind to the RNA after making the cDNA out of it. So these are similar to the DNA probe thing. You can also do RNA fish. So this is an example where people have used Jurkat cell line. Jurkat cell line is nothing but a T cell line, and they have tried to use look for three different RNAs. So you can do multiplexing also by using different colors. So it's just your imagination how much you can achieve. So this can help you to find out localization of, are those two proteins co-localized? Then obviously everywhere we are moving into automation. So the fluorescent microscopy is also undergoing a lot of automation. So this is a system which we also have, where you have a robotic system which will help you to stain your slides. So it has a platform where you can just put the slides, and there is an area where you put the buffers and your reagents. The robotic arm will move, will dilute the serum, put it on the slide, incubate, wash, put the next step, third step, and finally put a cover slip. Everything will be done. Then there is another thing here which you are seeing, which is a reader, which can read your slide. It doesn't need human beings now anymore. So you have this rack where the robotic arm will put the slides. They will go inside this system. One by one slide will be taken, scanned. It requires about 300 images for each slide. And because it has been trained by AI, that if you see this pattern, this is the interpretation. If you see this pattern, this is the interpretation. So the slides are read automatically by this microscope and you get a final report. So is it worth doing or not worth doing automation? This is the amount of time and cost you save. So this has been calculated. This is European costs, where the manpower cost is very high. So what they have calculated is that the total labor cost, hands-on time in minutes is 355 minutes if we use the current time for staining the slides for ANA. And if you use this system of automated system, you need only 82 minutes. So you can reduce your time so much, and that is why you can have cost saving. How good is it? So if you compare this with the current method, that is seen by a expert, the thing is about 85%. If you compare the technician reading the slide, not a trained, really trained manpower or a faculty reading the slides who have done it for 5-10 years, then even the technician has a agreement of about 89-90% for positive slides and about 95% for negative slides. If you use this machine compared to the technologies, it's fairly good. So where you don't have trained manpower, where the manpower is so expensive, and you have large volumes of slides, that is where automation is probably useful. In country like ours, where manpower is so cheap and our volumes are not large in each center because we don't do centralized things, it's probably not worth, because your infrastructure cost of buying that equipment is pretty high. We have bought it, but I feel that maybe we have not bought the microscope reading part of it. We just bought for automation of staining the slides, but even that is not cost effective. So just to summarize, that fluorescent microscopy is a versatile tool which has many applications. You can look for prefixed antigens in the tissues. You can look for antibodies in the serum. You can look at the DNA. You can look at the RNA. You can do for research, look for co-localization of proteins, many more things. And it has application in many fields, at least in clinical fields. The major application is in immunology, genetics, tumor biology, and microbiology. The most important thing which you need to standardize in the lab is the signal to noise ratio. That you should have good discrimination between negative and positives. Otherwise, your interpretation can be wrong. And you need trained manpower till the time the machines replace us. But even for that, you need to first have a good data set which is read by experts to train the machine using AI. You can never replace human beings. And probably in next 20 years, we'll also have automation in our own institution. Thank you.